Hi, Red Wolf. Hi, how you doing? So this is Dr. A, and we're gonna do a lesson on immunoassays. So I know most of you probably have already had immunology. If you've not, you will have it soon. But it's worth for us to talk about immunoassays because they are widely used in clinical chemistry. And they're immunoassay modules are a part of a lot of the bigger analyzers that are used in clinical chemistry. So start with a little review on antibodies, antigens, and analytes. So antibodies are produced against antigens, and this is in the body, and this happens in animals and all of that. The part of the antigen that is recognized by the antibody is called the epitope. So it's the little active site that the antibody has formed an attachment to, okay? And when we talk about antibody-antigen reactions, we have to talk about affinity and avidity, and those are specific to the antibody and epitope interactions. So, how well they bind and how strongly they bind. There's the bond is between the antigen and the antibody. And these interactions of these antigens and antibodies, which are predictable, then are the basis for all immunoassays. And then in immunoassays, what you have to know is that sometimes, like in this example, the antibody will be what will be part of the assay, the reagent. So for example, there's an antibody that's labeled here with something that can be detected. The antigen is what we're looking for as an analyte in a specimen. So it could be a hormone, it could be a drug level or something like that, or another protein. And if this is present, so this hormone or this drug is present, then it will interact with the antibody, and you would get an antibody analyte complex that is labeled and therefore will react with a color change, a fluorescent light or something like that. That can be detected, and then of course the higher the signal, usually the more of the analytes in the specimen. Okay, so your first Nearpod poll question for those of you on Nearpod, where's the epitope located? So answer that. Again, just kind of a basic review of antigen-antibody reactions. So some of the basics of the immunochemical reactions, so they do involve the binding of an antibody, which is usually IgG because it's more specific, to its complementary antigen, so that's the antigen that it was created against, okay? The antibodies can be monoclonal or polyclonal. Monocloning meaning that it comes from the same plasma cell line, so they have one cell that they've cloned, and that cell was activated against just that specific analyte that they were looking for, the specific antigen, and all it ever cranks out is the IgG to that specific antigen. Polyclonal ones come from different plasma cells, and so you have a little bit more variety in those, and so they could be two different epitopes and all of that. So generally, in most reagents, you're going to use monoclonal because it allows for more specificity. If you use polyclonal, you could have some cross-reaction. Antigens are proteins that are capable, or molecules, if you will, that are capable of reacting with an antibody. In chemistry, that would often be drugs that can be detected, but it can also be like vitamins, it could be hormones or something like that. An immunogen is a chemical substance that's capable of inducing an immune response, like a bacteria or a virus. So that can be slightly different in our understanding than an antigen. So an immunogen, if you get it in your body, if you get the bacteria in the body, and it's a pathogen even at that, because we don't even react against all bacteria, then you will make an immune response to it and create antibodies to it. When we create antibodies for reagents, they are created usually in a different species in reaction to something that would normally be present in your body that you would not normally have antibodies to. So if we're using an antigen-antibody reaction to detect vitamin D, it is not reasonable to think that normally vitamin D would cause you to have an immune reaction in your body. So vitamin D is not an immunogen, okay, but it is an antigen in this antigen-antibody reaction if you use an immunoassay to detect vitamin D, okay. And then a hapten is a molecule that's too small on its own to elicit a reaction with an antibody, and it has to be bound to a carrier protein to be able to stimulate an immune response and an antibody response, therefore, and also be able to bind to an antibody. A lot of drugs can be haptens, and so these are things to these are basic concepts to just remember. Okay, so haptens are too small, but on their own to elicit an immune response. Immunogens are the molecules that will elicit an immune response. So they're usually pathogens, bacterial and viral pathogens. An antigen is anything that's capable of reacting with an antibody, even if it's a normal molecule of your body. Okay, there are 5 classes of immunoglobulins. So antibodies are immunoglobulins. Immunoglobulins are antibodies. We use both terms interchangeably. There are 5 classes: IgG, IgA, IgM, IgE, and IgD. They each look slightly different. They all have a constant region, and it does have some biological activity, and it's the heavy chains of the molecule. So it's right here, it's the C part, C heavy, and a heavy chain. This end can bind to white cells and stuff like that. That's why it does have biological activity. And then there's a variable region, which is right here on the little arms, if you will, of your Y, arms of your Y. And the variable region is what makes it specific. It has light chains, which are the smaller light chains there, along with these heavy chains. And this is the part, a variable part is the part that is specifically made to an antigen, and it's made to fit like little puzzle pieces and to bind specifically to that antigen. And so it would not bind with another antigen because it just wouldn't fit. Okay, so now the concept of affinity and avidity, which I mentioned on the first slide. So those are related to antigen-antibody bonding. They are due to the electrostatic interactions, hydrogen bonds, and hydrophobic interactions and van der Waals forces between the antigen and the antibody. So there's quite a few different variables here that make an antigen bind to an antibody. Okay, so the affinity is related to the strength of the bond between the antigen and the antibody. So high affinity antibodies will bind more antigens. And the thermodynamic energy of interaction of a single antibody binding site plus its epitope on an antigen is always a property of the antigen. So does the antigen have the right shape for the antibody that was created? And so if it does, then it will bind with high affinity. If it doesn't, it might cross-react if it's similar, or it won't bind at all if it doesn't have the right shape. The avidity is the stability of the complex. So it's based on the affinity, so it's related to the affinity, and also on the number of binding sites, especially on that molecule that you're trying to detect. It is the overall strength of the binding of an antibody to an antigen, and it is always a property of the antibody. So it's how strongly that antibody locks onto that antigen and holds on to it. You do want, again, a good stability, good avidity in your immunoassays, because basically if you have labeled the antibody with something that's detectable, once it binds on the antigen, you want it to stay bound to that antigen until it is detected. Okay, you don't want it to just let go and be washed off. Okay, so which immunoassay characters represents the strength of the bond? Is it affinity or avidity? So it's a quick little memory test. Okay, so there's different big categories of immunoassays. So let's first talk about heterogeneous immunoassay. I can never say that word. Anyway, it's a mixing. Hetero means different, genus altogether. Okay, so heterogeneous immunoassays. They all require a separation step. So usually the separation step can be either a chemical precipitation, so you usually have a protein precipitating chemical. It could be an immunologic precipitation, so you would get a second antibody that would precipitate out the antigen-antibody complexes. Or you can have a liquid phase adsorption, so the free antigen is adsorbed or stuck onto like charcoal, for example. Or you can have a solid phase adsorption, and the binding will take place on the surface, solid surface, such as the wells of a microtiter well plate, or beads in the reagents. So again, heterogeneous, there's a separation step. So we're going to see a little bit how that works in just a minute. Homogeneous immunoassays, there's no separation step. And so everything is mixed in and nothing has to be separated out and can be just detected right there all together in the mix. This is usually used in urine tests for drugs of abuse and therapeutic drug assays, and basically a labeled antibody binds to the analyte, and the more analyte is present, the more antibody is bound and the stronger the signal is. Okay, so again, what's the difference, the big difference between homogeneous and heterogeneous immunoassays? All right, and then there's another classification of immunoassays, and that's going to be competitive and non-competitive immunoassays. And so again, you can mix, so you could have competitive and heterogeneous, or competitive and homogeneous, etc., etc. So it's just another way to classify them. Okay, so competition. In competitive immunoassay, you have competition between an unlabeled analyte from the sample and a labeled antigen that's part of the reagent. Okay, so for example, if you were trying to detect vitamin B12, you would there would be the vitamin B12 in your serum sample that is competing with a vitamin B12 in the reagent that is labeled with an enzyme or fluorescent molecule or something like that, and they are both competing for the capture antibody. Okay, and so what happens in this one, because they're both competing, is some of the capture antibody is going to bind your sample analyte, and some of it is going to bind your reagent analyte that's labeled. Okay, and in the end, basically, the more of the B12 in our example there is in your sample, the less of the reagent's B12 can bind to the antibodies, and therefore the lower the signal is. So in this one, it's inversely proportional. So a high amount of B12 in the sample would cause a low signal because only a few of the labeled reagent antigens, B12 antigens, can bind. Whereas a low level of B12 in your sample would then lead to a lot of the antigen from the reagent, the B12 that's labeled from the reagent, to bind on the capture antibodies and would cause a very strong signal. So that's the inverse relationship in competitive immunoassays. So there is a one-step format and a two-step format in your competitive immunoassays. So you have an antibody, an antigen that you're trying to detect, usually whatever's in the sample, and then an antigen that's labeled. The antigen that's labeled and the antibody are always part of the reagent. They're mixed up, and they're both competing again for the capture antibody, for the same antibody, and again that signal will be inversely proportional. So just like I explained with the vitamin B12. Okay, with a two-step format, you have first the antibody in excess is mixed with the antigen that you're trying to detect, and that capture antibody is going to capture all of those antigens, so all of those molecules, all of those like B12 molecules like we were talking about in our first example, and there will still be some leftover antibody, and then that leftover antibody is then going to, whatever's left can react with the labeled antigen with the signal. So you have, in the second step, you have these that have already bound with your sample, the antigen antibody from antigen from your sample bound with the antibody from the reagent, plus the excess reagent antibody that was left over. Now we add the labeled antigen, so the B12 that's labeled, and they will interact here with whatever's free of the antibody, and whatever's left then usually gets washed away and just gets trapped, and then again you read it, and so it will also be an inversely proportional type of signal, because obviously the more B12 you have, or the more of your antigen you have at the beginning, the more of the capture antibodies will be taken up, and the less you will have available to bind with the signal antigen. All right, so next little question. In a competitive immunoassay, then a high concentration of the analyte measured would yield a stronger signal or a weaker signal? Answer that. Okay, then you have non-competitive immunoassays. Those are also known as sandwich immunoassays. They do have a higher level of sensitivity and specificity, and they are always directly proportional. So they are sandwich because you make an antigen-antibody sandwich with the bread being the antibodies and the meat in the middle being the antigens. Okay, so usually the way it is you have a capture antibody that's usually fixed on a solid support. This can be microparticle beads, microtiter plates, or something like that. So they're fixed on there. If the antigen of interest is present, it will be captured on these capture antibodies, just right here. But then you need to be able to detect it, and so there's going to be an antibody to a different epitope on that antigen that is a labeled one that you then add. And if there is antigen bound here, then this antibody can bind on the antigen and then emit the signal, which is often a color change. The more antigens present that got captured, the more of the detectable antibodies will bind to it, and the stronger the signal. If there is no antigen at all present, then there's nothing for the signal antibodies to bind to, and there will be no signal at all. So it's directly proportional. The higher the concentration, the higher the signal. Okay, so this neuropod question, again, a non-competitive immunoassay, a high concentration of the analyte, then will yield, tell me, a strong or a weak signal. Okay, so since we've introduced the concept of labels, let's talk about all of these labels and what all can be used to label these antigens or antibodies. So again, labels are used as detectors in immunoassays. The labels are what give you the signal that you're measuring. Okay, an example would be a fluorescent label like fluorescein, and it will emit a light that can be detected using a phototube. There are also chemiluminescent labels. They're now very commonly used, like luminol or acrylium esters, and then these labels would interact with a chemical. There'd be a chemical reaction that would generate light. Radioisotopes can also be used. That is radioactive, so they would emit radiation type of signal that can be picked up. Enzymes are also very commonly used, like peroxidase and alkaline phosphatase. When enzymes are used as a label, they often just facilitate a substrate that has a chemical change that will change colors. There's usually a color change going on here. The immunoassay labels, so the labels are chemically bonded to the antigen or the antibody using a series of chemical reactions. That is done by the reagent manufacturers. It's not something that you would be involved in as a bench tech. And then, of course, the instrumentation that is used to detect a label will depend on the type of label used. If it is a color change, then it's probably going to be spectrophotometry, because you're looking at change in color, absorbance, and all of that kind of stuff, wavelength. If it's a chemiluminescent label, fluorescent label, then it's going to be like a phototube, just like detecting light intensity and stuff like that. If it's radioisotopes, it's going to be something to detect radioactivity. Okay, so yeah. That's determined by each manufacturer. Okay, so which of these immunoassay labels would you, with which of these immunoassay labels would you use a spectrophotometer? So I'll give you some examples of different immunoassay labels, see if you can figure out what's what, and answer me that. All right, next, which label would you expect to emit EMR? So electromagnetic radiation, which one would emit that? So let's look at the first specific example of immunoassays. Okay, so you have ELISA, Enzyme-Linked Immunosorbent Assay. So this is going to be a usually capture sandwich type antibody, antigen-antibody reaction, usually. So ELISA is a solid phase immunoassay. What happens is either the antigen or an antibody is adsorbed or fixed onto the little wells of the microtiter plate, and then the sample is added to form an antigen-antibody complex. So if you are trying to detect an antigen, then an antibody would be adsorbed onto the plate to capture that antigen. Okay. If you're trying to detect an antibody, then you could adsorb the antigen that it would react to onto the surface of the microtiter plate. Okay, so you might want to look if they have an antibody to something. Okay. Then after that initial reaction between whatever the antigen or antibody that has been bound to the plate has reacted with the antigen or antibody that is in the sample, then there's a wash step, and the wash step removes any unbound material. All that's gone, so whatever's left is whatever has bound to whatever to the antigen or antibody that was bound on the well. And then you add the enzyme labels, and they're added and washed again. Usually, so the enzyme labels are tagged onto an antibody, and then they react and either bind or don't, and then they're washed again. And then you add the substrate or whatever will cause the reaction. So this is an enzyme linked, so therefore the substrate will be the substrate for that enzyme. And generally speaking, ELISAs, what they do is a color change, and then you read the absorbance, and then the intensity of the color is proportional to the amount of either antigen or antibody we're trying to detect. So in a direct ELISA, you have an antigen that's added, that's bound to the plate, and then you have a detector antibody, primary antibody conjugate with an enzyme and a substrate and color change. With an indirect ELISA, you have an antigen again that is fixed on the plate. You have a first antibody that's right. This is what you're trying to detect in there, and then a second, an antibody to an antibody to get your signal. The sandwich ELISA, which is really the one that's used the most, you have the capture ELISA, the capture antibody that's bound on the plate. It captures the antigen it's looking for, and then there is another antibody that's added, and then there's an antibody to that antibody here that would cause a color change. And in competitive ELISA, then you have an inhibitor antigen that's competing with the other bound antigen, and both can emit the signal. So anyway, to give you some different examples of all the different ways you can just play with ELISA. So here, again, in the Enzyme-Linked Immunosorbent assay, the steps, you have the antibody that's, again, coated on the plate for this one. And if this is the indirect one, you add the sample, the antigen that's present is bound on that capture antibody, and then you add another antibody to the epitope, another epitope of the antigen, and then an antibody to that antibody that reacts and that has the signal, and then there you go. You get that enzyme that reacts with a substrate, which is the star here, and causes a color change that you detect. This video I will link in the bottom. This is a simple little video. I'm going to play it in class, but it does show how ELISA works, immunoassay work. It's very nice. Alright, so technicians in the hospital lab use an ELISA to measure rubella antibodies. They noticed that over the past 3 weeks, their controls values were consistently above the mean, and all values for the patients were high. So they're detecting more than what they intend to detect, right? There was also poor within assay precision. The lot numbers for the kids were all identical, so it's not a kit change. And the kits had been properly stored, so it's not a storage issue. What do you think is the explanation for these high values? So think about all the steps involved in the ELISA and put a possible answer there. And we'll discuss it in class. And if you want the answer from me, you can either ask a question on the video, or you can, if you're in class, just ask me during lab. All right, the second one is the IMIT. So it's called Enzyme-Multiplied Immunoassay Technology. It is used for drugs of abuse and therapeutic drug monitoring. It is a competitive binding process. And in this assay, the concentrations of the drug in the sample are proportional to the enzyme activity. So in this IMIT, you have part of the reagent, you have an enzyme with an analyte, antigen of the analyte that is bound on it. And then you have antibodies that are part of the reagent also. Okay. And so, these are your reagent, and you're going to incubate it with the sample. And so, if what you're trying to detect, the analyte you're trying to detect, is not present in the sample, it's just not there. So the drug of abuse, it's just not there. Okay. Then what's going to happen is these antibodies will be free to react with the antigen that's bound on the enzyme, and when they react, they're blocking the active site of the enzyme. So, and therefore, it's inactive, and the enzyme cannot interact with the substrate and cannot emit a signal. So basically meaning, if there is no antigen present in the sample, there will be no signal emitted. Okay. If the antigen is present in the sample, so the drug of abuse you're trying to detect is present in the sample, then what's going to happen is that antigen is going to soak up and it's going to pick up all these antibodies, and therefore those antibodies will not bind to the enzyme bound antigen, okay, in the reagents, and therefore the active site of that antigen will be, that the active site of that enzyme that's bound with that antigen will be available to react with the substrate, and so therefore the enzyme will react with the substrate and create a product. This should say substrate instead of enzyme, but so anyway, the active site is free. It reacts with the substrate and causes a color change, which can be detected. And of course, the more antigen is present, the more antibodies it will have bound, and the stronger the signal will be produced. So that one's a direct proportion. Okay, so in IMIT, again, the higher the enzyme activity, then the higher the concentration of the analyte, or the lower the concentration of the analyte. Okay, so Fluorescence Polarization Immunoassay, or FPIA, is a homogeneous competitive immunoassay. So everything happens in one reaction, one well, and there is competition between something that's labeled and then something that's in the sample. So you have antigen antibody fluorescein molecules will emit light in the same plane as the absorbed light energy, is the principle behind the fluorescence polarization. So you have an antibody in a reagent, you have an analyte fluorescein label in the reagents, and then you have the analyte in the specimen. Again, could be a drug, hormone, something like that you're trying to detect. Okay, so these two obviously are going to compete with binding to the antibody. So after the reaction happens, some of the you'll have some antibody antigen and some antibody labeled antigen, fluorescent labeled antigen complexes. Okay. Then what happens is you shine a light source at 420 nanometers, and the antibody antigen fluorescent rotates slowly in the solution, and then the signal goes through a polarized filter and can be detected. And if there is a lot of antigen antibody fluorescein molecules, then they will rotate slow enough that the light will be picked up through the polarized filter, and there will be a high level of measured fluorescence if there's a lot of these antibodies there that have bound onto the fluorescein label, which means that there would have been a low amount of antigen of the analyte you're trying to detect there in the solution. Okay, And so if there was a high amount of the analyte in the solution, then a lot of these fluorescein label antigens will not be bound to antibodies and would actually be spinning so fast that the light would not be detected through the polarized filter, and you would have a very low signal. So, and this is again an inversely proportional, whereas the least amount of the analyte is present in the solution, the less of it is competing, and the more of these fluorescein antigen antibodies are created, the more light's emitted, so the stronger the signal. The more of the analyte is in the solution here, the more there's in the solution, the more it's picked up all of these antibodies, and these antibodies cannot react with the fluorescein antigen and the reagents, and that light will just, that polarized light just will not be detected. Okay, this video, which I will link again on the bottom, I think is a decent illustration of how polarized immunoassays work. Okay, so then you have the microparticle enzyme immunoassay. It's called MEIA. And in that one, the surface of small beads are used to isolate these antigen antibody complexes. This is another one that's directly proportional, meaning the more analytes is present in the sample, the stronger your signal will be. So you start out with the reagents that are microparticles that are coated with an antibody to the analyte. Okay. And they're incubated with the sample, and the sample provides the analyte, if it's there, to form a reaction. Then part of this mix of reagent and sample will be picked up and transferred to a glass fiber matrix, which will trap these microparticles. And then an alkaline phosphatase labeled anti-analyte antibodies are allowed to then bind to the microparticle complex. So these little microparticles have captured some analyte, and so we're going to add an antibody to another epitope of that analyte, and it will emit the signal. So it's kind of a type of sandwich immunoassay, if you will. That's just, instead of being on a well, it's on a microparticle. Okay. And then you add your substrate to that enzyme, and you get a signal. The stronger the signal, the more the little antigens analyte there got trapped on the beads and sandwiched with the detector. So stronger the signal, more analytes in the sample. So again, an MIA, a high concentration of the analyte would yield strong signal, weak signal. We're just really playing with direct and indirect relationships. Then you have the chemiluminescent microparticle immunoassays. So same principle, except it's a chemiluminescent compound instead of an enzyme. Instead of triggering a color change, it would produce light. And so it's non-competitive, it's sandwich, it's heterogeneous. And it uses a magnet in the separation phase to magnetize the the microparticle beads that have sandwiched the antigen of detection. Obviously, you're going to use a photomultiplier tube for detection because you're trying to detect light, because it's a chemiluminescent assay. Again, chemiluminescent is a light reaction that's chemically based. It is one of the most sensitive assays, meaning it can pick up really, really low amounts of an analyte, which is really nice. And so in this one, again, it is directly proportional. The more light, because it's a sandwich immunoassay, so the more light is produced, the more analyte there was in the sample. So again, here it is. Illustrated, you have the little microparticle, you have your sample, and then you have your capture antibody, your reactant antibody here. Then your they're added in succession, right? And then your substrate here for the alkaline phosphatase that is on enzyme that's on this antibody. So. In the MIA here, the microparticle reacts with the sample. If there's enough deanalyte in the sample, obviously it's going to bind on these captured antibodies in the microparticle, and then you would add this alkaline phosphatase conjugate. It would go on the other side, the other epitope on the antigen, it would be right here, and then you add the MUP here that reacts with alkaline phosphatase and causes a color change. With the Chemiflex one, it's a magnetic microparticle sample, and then an acrydium derivative conjugate, which then, this is chemiluminescent, causes light change instead of a color change. And there's the chemical that would trigger the chemiluminescent reaction, and then you would detect light. So pretty much they're both microparticles. One of them is magnetized. They're both sandwich assays, just one of them uses enzyme color reaction and the other one uses chemiluminescent reaction. This other, again, video, I'm not going to play it. I will play it in class. I'm going to link it below. This shows also electrochemistry and chemiluminescence and stuff like that. Very interesting one, good illustration. And then you have the clone enzyme donor immunoassay, or CEDIA. It is another homogeneous enzyme immunoassay. So it uses an enzyme, it's homogeneous, everything the same well. It is used in genetic engineering techniques using E. coli. And the signal is directly proportional to the amount of analyte, and is often used for therapeutic drugs and drugs of abuse. So moving on from that one, so here's your more the explanation of how it works. So you have an enzyme acceptor, you have an antibody, and you have an enzyme donor. Those are all part right here. These three components are part of the reagents. And then you incubate it with a sample. If there's no antigen in the analytes, the antibodies are free to react with these enzyme donors, and the enzyme acceptors cannot react with the enzyme donors because they've been neutralized by the antibodies and they stay in an active form and there's no signal. If the antigen is present there, the analyte is present in the sample, then it will pick up all the antibodies, and therefore the enzyme donors can react with the enzyme acceptors and cause an enzymatic reaction, which would then cause a type of color change, whatever that's detected. Oh, a signal, basically. In CEDIA, the more analyte is present in the sample, the weaker the color or the stronger the color reaction, or signal if you want. In particle methods, it's based on the precipitation of antigen-antibody complexes. There are no labels, so they can be qualitative or quantitative. And your zone of equivalence in the antigen-antibody curve is important. So you don't want to be in antibody excess, which is the pre-zone, or in antigen excess, it's the pro-zone. Ideally for detection and precipitation, precipitation will happen in this antigen-antibody equivalence. We have just enough antigens and antibody in proportion. So out of those, you have immunodiffusions. So antigens and antibodies diffuse through agar. And this one, no electricity is used, just time. Radial immunodiffusion is an example of immunodiffusion, and you just incubate them and let them move and react all on their own. As the analyte diffuses into the gel, specific antibody will then bind and precipitate forms, and then you see these white circles of precipitates. The size of the precipitation ring will depend on the quantity of the analyte that was loaded, that was part of the sample. So there you go. So it's pretty simple. Slower method was used extensively. It's been replaced a lot by the automated immunoassays. Another type of immunodiffusion is the Occhialoni double immunodiffusion, also called the Laurel technique, which is called rocket immunoelectrophoresis. The antibody is in the agar and the samples are put in the wells. It is an adaptation of radioimmunodiffusion, except that instead of diffusion in a circle, it diffuses up in as a rocket, as peaks and stuff like that. So you have your calibrators, and again, they precipitate out in peaks like this. And you always set up all your patients in duplicates and stuff. So anyway, so it looks like little rockets, and so that's what's called rocket immunoelectrophoresis. Again, no labels, detectors, or anything like that. It's just precipitating into an agar, into a gel. And then, of course, you have immunoelectrophoresis, which is a two-step process. You first separate the proteins in the sample are separated by electrophoresis, and then an antisera is added to react with the separated antigens, and you get visible reactions. So the antisera obviously has to be specific to the antigens. In counter immunoelectrophoresis, it evaluates antigen-antibody binding, and they have to be oppositely charged. The Western blot is an example of an immunoelectrophoresis. It is widely used. This is how the Western blot works. So you have your infectious agent. For example, the Western blot was the test of choice to confirm HIV. So let's say you have your HIV, you have your lysate here, and then you have a polyacrylamide gel. You load your sample lysate in here with the different samples. You run your electrophoresis. So you have, this is just one band. You have your sample right here. The proteins migrate via charge and size across the gel. Because of their size and charge, it will stop and precipitate at different areas of the gel. Okay, then you take a natural cellulose paper and you blot it, or you sandwich it on top of the gel, right? And that picks up the proteins that were precipitated in the gel onto the paper, and then they react here with, you take them, you can cut them in strips, and then you can react them with the antisera antibodies to those protein bands. And then they bind there, and then you have antibodies to those antibodies, and they have the color, the enzyme that does the color change, and then you add the substrate, color change happens, and then you have colored bands. You can see the colored bands on the natural cellulose paper where the protein precipitated out. There you go. So that's Western blot. And then it's specific then, of course, for whatever it is that you're trying to detect. You know, in this case, we just decided, you know, HIV, because it's it was the old confirmation method of positive HIV tests. Okay, so light scattering methods. You can use turbidimetry and nephelometry. So these are widely used in your automated immunoassay analyzers, do big analyzers. Turbidimetry, if you remember in our instrumentation chapter, uses a spectrophotometer, and the more antigen antibody complexes happen in the sample, the cloudier the sample is, and it measures the amount of light transmitted, which is related to the concentration. The more concentrated it is, the cloudier it is, the less light is transmitted across the cuvette and detected on the other side. And then nephelometry is light scattered, so it's little particles of antigen and antibody that are in solution. As you hit them with light, they scatter light. And of course, for that one, the detector has to be placed at an angle, but it is more sensitive than turbidimetry because you can detect fewer particles that way. Again, there is no label in either one of those. It's just straight up just antigen-antibody reactions that are precipitating or not. Some of the factors that can affect immunoassay performance are going to be calibrators. You have to handle your calibrators properly and establish a dose-response curve. There are differences between manufacturers, and one of the biggest things is that what you need to take away is an immunoassay done on one analyzer with one manufacturer's kit cannot be compared with another immunoassay done on a different analyzer with a different manufacturer's kits. I would even say that from one facility to the next, one analyzer to the next, you probably can't compare them either. So they're very specific to they're by manufacturer, by brand, etc., etc. So you have to obviously establish your signal calibration line. You do that by running known concentrations of your immunoassay, your analytes, and you get your signal detection, plotting against the signal detection, which could be absorbance or light emitted or whatever. And then that's will establish your calibration line. So these are your, using standards. And then all your unknowns are your samples. You would get a specific signal that would correlate then to a concentration based on your calibration line. You have to run controls. Controls check the performance of this calibration line at the low, middle, and high end. Okay, so there's usually 3 levels of controls. Those have to be checked at least, I would say once a day for sure, some of them once a shift. There are things that can interfere with assays. So, for example, the presence of heterophile antibodies, they can bind to the assay antibody and keep it from reacting with the antigen, etc. So they could cause basically false low readings on some samples. And then there's the high dose hook effect, which is basically the prozone effect, where whatever you're trying to detect is in such high amounts that it's crowding out all the signal and everything like that. And so here's the hook effect. And so you need your excess detection antibody. You have to have enough antibody to get a positive slope in this dose response curve. If the analyte is present in excess, the curve can flatten out and become then negatively sloped, which is making this hook here. And so if you have a sample where you would have expected to have a higher result, but instead you're getting questionable results or low or no readings and stuff like that, what you have to do, which is kind of weird because you're getting a low signal, but you were expecting to have a high one, then you have to dilute it because you have so much antigen that you don't have your excess antibody. So you need to dilute it to where you get back to where there's excess antibody in your reagent. And so you dilute it out to get a stronger signal, which is kind of counterintuitive here if you understand dilutions and stuff. So if you do not confirm this abnormal reading, this questionable results with a dilution, you could have a severe underestimation of the true analyte concentration in these samples. So it's something to remember for immunoassays, is they are subject to the hook effect. Okay, and then if you have any questions for me in the Nearopod, feel free to put them in there or send me an email if you're in my class. And thank you for your attention.